Hi, everyone. So before we start with our lecture today, just a quick couple of assignment reminders. So you have your first real quiz due tomorrow night, Thursday night by 11:55. I know last week on Friday, I said, oh, maybe I'll just open up the quiz on Friday. And if you want to take it, you can take it. But then I thought about it. I was like, that doesn't really make much sense because a couple of the questions on the quiz have to do with stuff that we'll cover today. Right. So it's better that we waited until we go ahead and went over it or go over it in order to put everybody in a good position to be able to take the quiz. As a reminder, please keep your mics muted during the lecture and please turn off your video during lecture as well. Otherwise, I always have to get distracted and shut down your webcam for you. Okay. All right. So you have your first quiz. It will automatically open up at 1:30 today. So after our lecture is over, you'll see quiz number 1 appear. And you will have until tomorrow night, Thursday night, 11:55 to take it. All right. If you haven't done so already, I recommend you to take the practice quiz because the practice quiz is just like the real quiz, right? So it's sort of a way of test driving it to see the nature of the questions and the whole experience. Importantly, you actually have your first inquisitive homework set due tonight, Wednesday night. So I should have mentioned this first, right? Because now I'm like going back in time. Your first inquisitive homework set is due tonight by midnight, and that's on inquisitive activities 1 and 2, the first 2 chapters of the book. Okay, so yes, they do take time. I think on average, it takes somewhere around 40 minutes to do each one of them. For most weeks during the semester, your inquisitive assignment only covers 1 chapter. Right? But for this first week, we were doing 2 of them because the first chapter is just kind of an intro anyhow. All right, so don't forget to do that. Get your inquisitives done tonight and remember to take your quiz by tomorrow night once the quiz opens up in La Lima after today's lecture is over. All right, okay, great. The other thing I wanted to mention before I forgot, I think all of you are aware that UH has extended the period for online teaching now all the way through next week, and classes will start in-person meetings on Monday, January 31st. So even though UH is giving faculty the option of whether to stick with online for next week or actually make students come back for in-person next week, I think it's better we just stick with online for 1 more week. Okay, it's a bummer because I actually do really look forward to seeing all of you in the lecture hall, right? Because I always look forward to that and interacting with you. But I think to keep things easier, let's continue doing online classes through next week. And that way, when all classes resume in-person classes on Monday, January 31st, that's when we'll actually also switch back. And I'll look forward to seeing you in person at that time. The exception is for those of you who are taking the lab class, right, the 461L. We had already delayed the start of the semester by a couple weeks, and we can't really afford to delay it any further. So next week, we are still sticking with our plan to begin our in-person lab classes on Wednesday. Okay, I think those of you who are taking the lab already saw that announcement in La Lima. All right. So enough with the administrative stuff. Let's talk about immunology. Quick review, right? We've learned as part of our introduction to the immune system that the human immune system consists of 2 main arms, our innate immunity, right, and our adaptive immunity. And we've been focusing thus far on the innate immunity because this is the part of our immune system that comes pre-programmed in all of our genes, right? In other words, the know-how to make certain cells that act as effector cells that are patrolling around and looking for common everyday pathogens, and the know-how to make proteins like complement proteins that can opsonize or attack pathogens, coating them, making them easier for things like macrophages to eat. That's all part of your innate system, right? Our adaptive system is the side that has to deal with brand new threats that maybe come along and our body has never seen before, or maybe I always say brand new to the whole human species, right? We have to have a way of adapting to that and dealing with it. Otherwise, quite simply, right? This is bad clown, right? If bad clown comes along and your immune system can't recognize and eventually deal with them, that's the end of the line for you, right? You get killed by bad clown, right? So adaptive immunity, we're going to talk about soon. But today, we're going to sort of continue talking about the innate side. Right? And even within the innate side, remember, there's two sort of stages of how your innate system works. You have the immediate response to pathogens that get into your cells, into your body's tissues. So, for example, macrophages, complement proteins, right? They are all part of the immediate response because they're already there. Okay. And there is what we call the induced side of our innate system. In other words, if those macrophages and complement proteins can't take care of the threat right away, or maybe even within a few hours, right? And let's say a day later, there's still bacteria in there, right? Then they have to call for backup, right? And the backup is your induced side of your innate immunity. So today we'll finish talking about the immediate side and start transitioning into the induced side. And that's what we'll continue talking about next time. All right. So we had introduced complement proteins as being these proteins that are made by your liver, primarily made by your liver. And you have tons of these complement proteins floating around in your blood, and they're seeping out of your blood into your tissues. And we said that complement proteins are a great example of very important effector components of your innate immune system that are not actually cells, right? These are just teeny tiny individual proteins, right, that have evolved to be able to activate and attach to the surfaces of pathogens, thereby labeling through coating the surface of the pathogen, a process that we said is called opsonization, right? And that coating can do things like make it way easier now for macrophages to come along and stick to the bacteria, right, and eat it up. Right? So we said last time that there are three pathways through which complement C3 can be activated. Remember, complement proteins is a family of many different complement proteins, but C3 is probably the most important, right? And last time we looked at those really scary pathways of how C3 activates and attaches. So what we looked at last time was known as the alternative pathway of C3 activation. Right? It is the first one to act. Right? So it's part of your immediate innate immune system. And so the pathogen surface creates a local environment that's conducive to the complement proteins activating, and then they stick to the surface. And once they stick to the surface, right, they become these convertase complexes by recruiting other complement proteins. And then that really now amplifies more C3 being activated and stuck to the surface. Right? We will learn more about these other pathways, the lectin pathway and the classical pathway, in due time. But let's just continue with our discussion of the alternative pathway and the eventual activation of complement C3. Right? So remember, I already warned you that the cartoons in the book, while they're nice, you know, because they're very simple and try to help you focus on sort of conceptually how these different cells or proteins are interacting, they're kind of dumbed down to the point where they can be misleading, right? For example, the complement protein we've seen previously is just represented as a blue egg, right? And I said, don't any of you believe there's actually this blue egg. So I want to take a moment, right, over the course of a couple slides and just remind you, right, some basics about protein structure. So this is all a rehash kind of Bio 275. For those of you who have taken it already, this should be review. For those of you who aren't familiar with this, hopefully this will give you enough of an introduction. Different ways of visualizing protein structure. Right. So in many of the book's cartoons, something like complement C3 is just represented as this blue egg, right? And this pair of scissors comes along and cuts the blue egg. And now we said inside the complement protein, there's a thioester bond that then is exposed. And that thioester bond here will really strongly interact with some other molecule that might be on the surface of, let's say, a pathogen, right? So on the surface of this bacteria, generically, let's say there's some sort of R molecule, right? Then this complement protein can interact with that. And we learned that in the process of activating, right, this C3b then becomes attached to the surface of the bacteria, right? The part that got cut off, the C3a, floats off, right? So this is a very functional, simple geometric shape kind of diagram. It gets the point across, but it can be misleading. So let's have a more realistic view of this here. All right, one way of thinking about protein structure, of course, is the primary sequence of amino acids that actually make up a protein like this complement protein. Right? So what we have here is this colorful map representation is if you unwound the complement protein C3, right? So instead of it being all balled up in its normal 3D shape, you unwound it, you would find, of course, that it's one continuous polypeptide chain of about 1,600 amino acids, right? Starting from amino acid 1 over here all the way to the end. So what they've done is they've color-coded the different regions of this protein that once the protein folds up in its three-dimensional shape, these are the different regions with particular amino acid sequences that have different functions. All right, to start off with, let's start from the very start of the polypeptide chain. So you have a methionine there, right? So this is the first 100 amino acid residues that might be found over here. Okay. Remember, these are the single letter abbreviations of amino acids, right? So again, if you're not familiar with this, look it up, okay? Because this is what you learn in intro cell biology. And you're like, what? I didn't know I actually need to remember this. Well, you don't have to remember them in terms of which one stands for which protein, but you need to realize and understand what this is representing, right? A chain of amino acids here. The first 100 or so make up this region, which we call a macroglobulin module. Right? So what you would find is that for the first almost 800 amino acids, right, it's just a repeat of these same 100 or so amino acids over and over and over again, right? Not surprisingly, this would probably lead you to believe that, oh, this particular shape of each of these macroglobulin molecules evolutionarily, right, is used for many different types of proteins that are involved in immune functions. That's how Mother Nature works, right? In the course of evolution, if Mother Nature stumbles upon a particular sequence of amino acids that makes a desirable shape that's useful, right, then that underlying DNA sequence that codes for that protein, you'll see it over and over again in different genes that might have related functions. Right? Then over here, you see you have a region that this thing kind of looks like a choo-choo train, right? But over here, the smokestack is this ANA, this blue triangle, right? So this is the actual C3a region of the protein, the one that up here, right, floats away when the protein is cleaved. Then you have this TED region, which is the thioester bond domain, right? So in here would be the hiding thioester bond, right? That's formed by two cysteine residues. Their side chains are interacting with each other to form this thioester. And then you have this pink region, which is the C-terminal domain. All right, great. Right, so that's a simpler way of thinking about the structure of this protein and the different parts of it that are going to contribute to its function. But, I mean, this is very misleading also, right? Because great, we've gone from a blue egg to now a multicolored choo-choo train, right? Okay, no. Time to grow up. Right? We're in immunology class. The reality is this whole thing folds up into 3 dimensions, right? So here you have the same color coding as you see down here, but now it's balled up in its normal 3-dimensional conformation. And you begin to see where these critical components are. Here's the ANA or the C3a region over here. Here is the thioester bond region. And here in the little, I think, orange triangle is the actual thioester bond hidden or tucked away inside of the inactive C3 complement protein. When the C3 is cleaved or activated, the blue ANA triangle will float away, right? So you no longer see it here. Right? But look what happens. Because that region has been cleaved, the rest of the protein adjusts, right? Because suddenly all the amino acid side chains, right, they're no longer happy in the position they were before. And so the whole thing begins to move around. And look at what happens. This is really cool, right? This domain, the thioester bond containing domain, naturally then ends up pointing down and out of the protein. Right? So now the thioester bond is pointing out is it is exposed, right? Just like you see over here. And now it can interact and form a covalent bond with whatever hapless target happens to be close by. So even more realistically, right, if we look at the ribbon diagram, remember this is all one polypeptide chain, right? But now it's still maintaining the same color coding. So you can see how these different domains are arranged here. And again, the thioester bond is tucked inside normally, but when the ANA region, this light blue region, is cut off, right, the whole rest of the protein kind of collapses and the thioester bond points down and out, right? So this type of ribbon diagram is very useful because now you're really beginning to see what this whole blue egg in this first cartoon really looks like, right? And the very last way of looking at it is if you now fill in that ribbon diagram with all the atoms, right, that represent all the amino acids and their various side chains. So this is really what the complement C3 looks like, right? And here you see, here's the ANA region, the red region that once that gets cut off, it floats away. The rest of it kind of collapses and the thioester bond now is sticking down. I think this is pretty amazing, right? And remember, this is just all a function of this long polypeptide chain having the right amino acid sequence so that it folds up in just the right way. And importantly, when it is modified by having part of it cut away, right, the rest of it through evolution, right, collapses to expose the thioester bond down below. So throughout the semester, I want you to keep this in mind. When we look at diagrams that are just very simple geometric shapes, right, always be kind of cognizant, right, that the reality is most of these are these very complicated three-dimensional protein structures, right? Okay, good. So let's continue talking about the role of complement proteins like C3. All right, so here we have what we talked about last time. When the C3 complement is activated, you're left with a C3b with a thioester bond that now, in this case, has successfully attached to the surface of a pathogen, like a bacteria, right? And it has recruited factor B, right? And now factor B has been cleaved. And so what you're left with is this complex of a C3b with a BB, right? And we call this a C3 convertase complex. Why? Because now it functions as a machine to convert other C3 molecules that are floating by into C3bs themselves, right? So this is the pathway that we looked at last time. This C3b BB, right, now that it's lost the BA part, right, it now can attract another C3 that's floating by. That C3 attaches, right, to this convertase complex, and the BB is a proteolytic enzyme complex, or not, it's a proteolytic enzyme, so it has a catalytic site that then cleaves, right? It represents the pair of scissors that then cuts this C3, right? And this C3 becomes a C3A that floats off and a C3b that's left over that hopefully is going to get plopped onto the surface of the bacteria, right? Once it comes off and gets plopped on, this original C3b BB complex, right, essentially goes back to being like this, and it can attract another C3 and over and over and over again. Keeping in mind that each one of these C3bs that got planted on the surface themselves can recruit their own yellow factor B, and themselves can each become a convertase complex. So very quickly, right, you get this rapid exponential coating of the bacteria surface with this complement protein C3b, right, which serves as the Velcro, so that now the beach ball has an easier time sticking to the Velcro labeled ping pong balls. But this is an interesting twist now, right? What if in this brief amount of time when this C3 convertase complex, right, has cut this other molecule of C3, for a brief moment of time, it's still hanging on to that C3b, right, before the C3b falls off and gets plopped onto the surface. Right? In that brief moment of time, another option can happen. So it can go this way. If this C3b, right, C3b, BB, right, is still hanging on to that C3b that it just made, right, in that brief moment of time, this shape actually is the favored shape to bind complement protein C5. So C5 might come in here, bind there, and then the factor BB over here goes ahead and cuts that into, surprisingly, right, C5a, which floats off, and C5b, which floats off. Okay? So why would it do that? Right? Well, because this C5b is now going to head off and be involved in a different pathway that helps complement proteins directly attack this bacteria. Right? So keep in mind this or. It means at this moment when this C3b Bb has bound to a C3 and cleaved it, right? It might be that that C3b falls off and continues the opsonization process, or by chance, if a C5 happens to be floating by in that brief moment of time, right, the C5 might be the thing that gets cleaved and floats off as a C5b. Right? Keep in mind that as this process is happening, you're going to get hundreds of thousands, if not millions, of individual C3bs on the surface of the bacteria, each one becoming little C3 convertase complexes, right? So even though this pathway is a this way or that way, the fact that you have millions of these doing either this way or that way at the given time means that essentially both of these pathways are happening at the same time, right? As far as the overall surface of the pathogen is concerned, right? All right. So what's the significance of this C5b? Right? So the C5b is involved in directly attacking and destroying the pathogen. How does it do that? Here you have an image of bacteria. Right? So you can think of bacteria just like you can think of any living cell as basically a sack of water and biological macromolecules, right? Sort of like this sack, plastic sack full of water here, right? So if you wanted to somehow disrupt the normal functioning of this bacteria, right, what might be a very obvious way of interfering with the normal functioning of the cell? Right? Well, you might say, what if we just like poked holes in it, right? Yeah, absolutely. If you can poke holes in the surface of the bacterial cell, then the cell can't function anymore. It literally blows up, spills all the guts out, and that's the end of the bacteria, right? Hopefully it's not the end of our friend the fish over here. Okay, but you get the point. So that's what the C5bs begin doing. So remember, the C5b, right, has been converted by this C3b I know, terrible name, right? They should have called it Fred or something, right? But anyhow, but at least this you can kind of piece it together because it's a C3b, right? 2 with a BB attached to it, right? It converts the C5 into C5b, and this C5b now floats off and starts forming what we call a membrane attack complex, or MAC. Right? So how does this happen? This C5b begins to recruit other complement proteins, C6, 7, 8, and 9, to start working together. Okay? So if you look here, the C5b associates with a C6 that's been floating around and a C7. Together, that puts them in the right shape to now get planted onto the surface of the pathogen in the lipid bilayer. Right? Once it's planted there, it's the right shape to recruit complement C8. And C8 really embeds and anchors this complex to the membrane. Right? Complement C9 comes along, and multiple C9s get added and actually forms a ring starting from this original C5, 6, 7, and 8 complex. Right? It adds additional C9s until it forms a ring or a tunnel in the membrane of the cell. Right? And you can see here this wonderful electron micrograph image. Right? This is the surface of a bacteria where these membrane attack complexes have formed. And it looks like Swiss cheese. Right? So as a result, yeah, the bacteria can't deal with this. Right? Water starts flowing in uncontrollably. Solutes inside the bacteria start flowing out uncontrollably, and essentially the bacteria is going to swell and burst because of everything, all the water being losing control over the water. Okay. Oh, here's a question. How big are the pores created by MAC? They're very tiny, right? They're like on the, sub 1 micrometer scale. So you're talking about nanometers in size across, right? Especially when you think about it, these are individual small proteins that are making the wall of it, right? So they're not that big. Okay. Don't be intimidated by this, right? I don't expect you to somehow draw this, but the truth is it's not that hard to know, right? That C5b that came from the original C3, right, that made a convertase complex and was able to convert C5, so it actually able to function as a C5 convertase and make this C5b, just sequentially recruits C6, 7, 8, and a whole bunch of C9 to make one of these membrane attack complexes. Right? So this is a very good example of how something like a dumb little protein or a group of dumb little proteins, right, can actually attack and destroy a bacteria without the help of a big immune cell coming in and phagocytosing the bacteria, right? So as I said, opsonization of the surface of a bacteria with C3, right, ends up resulting in both. It results in coating the surface of the bacteria with Velcro so that a macrophage can come in and have an easier time eating it. But at the same time, it's also coating the surface with all these little holes, right? And so that's a real powerful 1-2 punch to take care of things. I'm never satisfied just showing you the dumbed down little ovals, little eggs and things. So this is the actual sequence of events that we just looked at, but now with these wonderful representations of the true three-dimensional shape of these proteins, right? So again, I don't expect you to be able to necessarily recognize any of these shapes, but I want you to be familiar with this chain of events that happens. And I want you to always have the opportunity, right, to step back and appreciate the fact that, yeah, right, each of these are unique three-dimensional protein structures that all work together, right? You can really see that. Look how nicely they fit together, right? Like the light blue one here, that's just kind of like nestled right in the groove of this greenish one, right? That's how they stay together just through protein-protein interactions to form the right shape that ultimately then recruits a whole bunch of C9s to form this MAC complex. All right. So because this is mediated just by dumb proteins, proteins don't know what they're doing, right? They have no idea what they're doing. They just do what they do because of their shape. So this is potentially really scary, because what if your own cells in your body started having these membrane attack complexes forming on them, right? Because after all, if the bacteria got into your body, and this is where this whole battle of your immune system against the bacteria is taking place, right? You've got lots of innocent bystanders, like spectators, right? Which are your normal cells that are surrounding that area of the tissue, right? And they need to somehow be spared from this whole attack taking place, right? So let's imagine that a membrane attack complex starts forming improperly on a human cell, right? And this does happen, right? Because any cells that are nearby might start being accidentally, targeted by your complement proteins. So this complex of C5b, 6, 7, and 8 have already formed, right? All that's left now is to recruit the C9 to form a big ring and carve a hole in the surface. Fortunately, our cells evolved to have other proteins that discourage this from happening. So CD59, right, this yellow or orange one here, right, is the right shape to come over, because it's been on the surface of our own cells. It comes over and attaches to this forming MAC complex, right? And it puts an end to the recruitment of C9. So therefore, CD59 protects your own cells from having membrane attack complexes formed, right? An example of what can happen if you don't have this are, certain people might be born with genetic variations, alleles carried in their genes that might lead to a defective or misshapen CD59, a protein that doesn't properly associate with membrane attack complexes. Right? So in a normal red blood cell, for example, right, on the surface, you would have sort of this CD59 protein here, right? I know it's drawn as this funny purple sort of thing over here, whereas it's drawn as orange over here, but whatever, right? But some people might not have CD59. And as a result, their red blood cells might be very prone to undergoing attack by membrane attack complexes, complement proteins poking holes in them. leading to conditions where people just can't maintain normal numbers of red blood cells. They're always rupturing, right? So we call this a condition of hemoglobinuria, right, which can lead to anemia, right? Because with fewer red blood cells, you have fewer oxygen that's being carried around, and all of the associated symptoms of anemia, like weakness, fatigue, etc. Right? So this is just to highlight the fact, right, that many of these immediate, or I should say, innate immune functions that are carried out just by proteins, right, have to be tightly regulated, right? Because we don't want is, oh boy, you got to control a Big Mac attack, right? Yeah, I know. You know what? I every semester I tell myself this is the last time I use this slide because it's just really a pretty stupid slide, right? But the point, I can't get myself to do that because I spent like 2 hours making this slide semesters ago, so I always use it. All right. Okay. Anyhow, so let's go back. We said that our innate side of our immune system, right, has two lines of defense: the immediate response, right, that involves things like macrophages that are already there in the tissue that phagocytose and destroy bacteria. We also have learned now that there are certain proteins that contribute in this effort to fight against the bacteria or the pathogen, right, that don't even involve necessarily depending upon another immune cell to actually come along and do something. So we already looked at complement proteins. Now let's just quickly look at two other examples: macroglobulins and defensins. So these are proteins that you have right now in the tissues of your body, right? And they're just ready to meet up with any kind of bacteria or other pathogen that comes in. So macroglobulins are big proteins, right? And they are immediate bacterial inhibitors. So one way that bacteria colonize our tissues is once a bacteria finds itself in a favorable tissue environment, surrounded by wonderful nutrients, right, and it's all happy and it wants to start going nuts and multiplying now, right, it needs to kind of condition its surroundings, right? So bacteria do that in the same way that if you move into a brand new apartment, right, that has no furniture, nothing at all, just bare walls and windows, right, one of the things that you do before you get comfortable is to kind of like make it your own, right? You maybe put up some artwork, move in some furniture, whatever, and then you get happy, right? So bacteria do that as well. One thing that bacteria often do is they secrete different types of proteases. So proteases are proteins that cut up other proteins, right? So the bacteria is basically trying to carve out, no pun intended, right, a happy home in your tissue by digesting away surrounding tissues to benefit from, whatever raw materials that freeze up, right? So not surprisingly, your innate, immediate part of your immune system evolved to be very sensitive to that, right? And wanting to get rid of these bacterially secreted proteases. Right? So our immune system responds with many types of protease inhibitors. And so alpha macroglobulin is a good example of that. Right? So here you see in this cartoon here, right, the alpha macroglobulin is this big pink, I don't know what it looks like. It just looks like sort of a, room divider or something like that. Okay. And here you have the green Pac-Man, right, is the protease that's been secreted by the bacteria. Right? And it's going to damage your tissues if you don't neutralize this green Pac-Man, right? And so the macroglobulin is very cleverly evolved to take care of neutralizing the protease. How? Two components of the macroglobulin structure are that it has a thioester bond available to try to attach covalently to some target, and it has this bait region, right? So this bait region, this little ring, it's just part of the polypeptide chain. It's kind of like the bait region on like a mouse trap, right? The mouse trap, you bend the hinged actual, part of the trap back, right? Hopefully you don't snap your own thumb in the process, right? And then you have the little bait part where you put the little wedge of cheese or whatever, right? And that then, if the mouse comes along and touches that, right, that triggers the trap to snap, right? Amazingly, that's exactly what happens, right, with this protein. You see here the bacterial secreted protease, right, fell for the bait, right? And so it attached and cleaves now this bait region, right? The protease goes ahead and breaks the protein there, and that releases the spring, and the whole macroglobulin now snaps shut. And in the process of snapping shut, the thioester bond is jammed up against the protease and forms a permanent bond there, right? So that means this individual protease molecule now has now been neutralized, right? It can no longer harm any of your cells in your body. So yeah, this pink dividing screen thing, right? Perfect example of a sort of a figure that's a little bit too dumbed down for my liking, right? So you didn't think I was not going to let you see the real structure of this, right? So here is the actual structure of the macroglobulin. So you can see it's this large multi-subunit protein holoenzyme complex. In the middle here, you have this bait region, right? So if you were to fill in this shape, so this is just looking at the left side, right, of this macroglobulin, right? Here is the bait region, and here is the thioester bond region. So if this bait region gets cut, right, then the whole thing will now close up. And I said, when you look at this ribbon diagram, I don't know about you, right? But right away, if I imagine this thing closing up around, a target, to me, it looks like a bird of prey, right? And I think it's amazing when you think, right, that this thing functions like that. And all it is a protein that your genes, right, that you evolved with know how to make, right, to code for, and they can actually have these complicated functions. Right. Another example are defensins. So defensins are actually way simpler than a macroglobulin. And in fact, they're way simpler than even a complement protein. Right. So here you have a defensin molecule. So it's a short 35 to 40 amino acid protein that has amphipathic properties. What do I mean by that? Based upon the amino acid residue sequence, as you go through this short 35 or 40 amino acids, right, you would have more hydrophilic residues in the yellow region and more hydrophobic residues in the green region. So that's what gives an individual defensin molecule these amphipathic properties. Remember, amphipathic means that it both likes and also dislikes water. Right? That's why amphibians, when we talk about frogs and things, right? Amphibians can handle being in water and on land, right? So they're amphibians. So these defensins have that sort of property also. They both like water on one part and hate water on the other part. And so these defensin molecules then, they insert themselves into the membrane of bacteria. So here's the lipid bilayer. The hydrophobic part of the defensin molecule will naturally get shoved into the membrane because it's hydrophobic, whereas the hydrophilic part, the yellow part, happily associates with water molecules, thereby keeping this defensin molecule kind of happily embedded that way. So once you get several defensin molecules in the membrane, they will naturally, through their shape, attach to each other. And this essentially then pinches off a pore in the membrane, right? So this is way simpler than the membrane attack complex that we learned about 2 slides ago, right? So you say, well, yeah, if this is way simpler, then how come we just don't do this all the time? Because this is so simple, because it's literally just defensin molecules interacting the way that, hydrophilic water tension and electrostatic interactions are driving them. There's not a good opportunity here to actually control this, right? So that if your cells in your body were exposed to defensins all the time, you wouldn't have much opportunity, right, to actually protect your own cells of your body against this mechanism. That's why defensins are reserved for use only in certain situations. So for example, defensins are found in the mucus layers of our body, where you don't have many of your body cells present, but you certainly can expect to be trapping a lot of bacteria and stuff like that. Also, in different fluids that are secreted. So for example, in our sweat on our outer skin or tears, right, have defensins in them. Because again, out on the outer surface of your skin, you don't worry about these defensins actually attacking your living cells underneath, right, your outer layer of keratinized skin. All right. So those are just some examples then of additional immediate response proteins, right, that are working with complement to carry out an immediate response. They're already in you. They're ready to go. The moment you get poked and your skin gets kind of broken by a pin or something like that, and bacteria gets in, right, that bacteria is going to be subjected to these protein effectors. And at the same time, they're going to be subjected to whatever innate immediate effector cells like macrophages happen to be there, right? All right. So that takes care, of many infections, right? Like I said, if you get poked by a pin and there's several thousand bacteria that get in, right, that can get taken care of by these immediate responses within a few hours, right? So even though you jabbed yourself and you're like, right, chances are by the end of the day, you even forgot that you got poked by a needle, right? And you don't ever get bothered by it again. Right? But what if the amount of bacteria that got in was too much for these immediate responses that happen to be in the area to take care of? Therefore, after a few hours, maybe there's still bacteria and the bacteria is dividing, right? So now maybe you're talking about a day later. Oh boy, right? You better call for some backup help. Fortunately, we still have other innate components, right, that can come as part of the backup. And those would be our induced responses now. So maybe like a day later, right? How do these get called for? They're getting called for by those cytokine messages that the macrophages have been busy sending out, right? Ever since the first moment that the macrophages have detected that there's bacteria, either by eating the bacteria or by receptors on the macrophages picking up on different things that are secreted by the bacteria, right? Or by the increasing levels, right, of activated complement proteins, having things like the C3a parts of activated complement proteins that are floating off, right? These act as signals. Right? So the macrophage has been busy generating cytokine proteins, right? And these levels are beginning to build up. If after a few hours, all the bacteria is gone and the macrophages stop detecting any more bacteria, that means the macrophages stop producing cytokines, right? At the same time, if there's no more bacteria, you're going to suddenly find that you have fewer and fewer and fewer complement protein molecules that are actually activating. And therefore, you get lower and lower levels of small little C3a messages floating out as well, right? Or C5a as well, right? So that would be the end of it. But if you find yourself a day later, right, and these cytokine messages and complement signaling messages keep going up and up and up, that's then when they've now gone into your blood and they're flowing around your blood, and they can start attracting other cells to help out. So cytokines released by macrophages, the complements that are being activated and releasing C3a and C5a, these begin to stimulate changes in the local physiology. Right? The tissue begins to get inflamed, right? Meaning it begins to get swollen, it begins to get a little bit more red, it gets warmer, right? And of course, and it can also even get kind of, firmer, right? I think you all know what that feels like. If you get a cut and then a couple of days later, you're looking at it and like, ooh, right? It's getting a little puffy there, a little red and sensitive, right? Remember, up until taking this class, that's probably when you started saying, oh man, my cut got infected, right? When in reality, now you know. No, the infection technically happened at the very moment that you got that cut and the bacteria got in, right? It's just that this particular infection wasn't resolved or neutralized within the first few hours. And now a couple of days later, you're seeing the next phase, right, of your innate immune system beginning to start up. And that involves inflammation. Right? We'll talk about the functions of inflammation next time. But importantly, it also recruits, these cytokine messages recruit things like more monocytes. Remember, the monocytes are the precursors of the macrophages. The monocytes are the form of macrophages that are released by your bone marrow. And then these monocytes are circulating around in your blood. And when they get to somewhere where they're needed, like here, right, the site of infection, and they begin to sense all these cytokine messages, they will then squeeze out from the wall of your capillary, right? Because remember, this is a thin little capillary here. The white part is the inside of your capillary. And so cells like this monocyte can squeeze out and become additional macrophages, right? Or they can become dendritic cells, which we'll learn about a little bit later. And neutrophils also get recruited, right? And neutrophils, as we'll learn next time, are really potent phagocytic cells, right? Very potent phagocytes that help out the macrophages to eat even more bacteria. Okay. So that's why I'm saying here, as a reminder, that in addition to the cytokine messages that are being released by the macrophages in response to them detecting that they're eating bacteria or detecting different components of bacteria by way of different receptors on their surface, right? The C3a and C5a that are being released by activated complement proteins, right? They also serve as messages to recruit other cells, right? So when these were first described, they were called anaphylatoxins. Right? You might have heard of this term anaphylactic shock, right? So anaphylactic shock can happen, for example, if somebody is deathly allergic to, peanuts or bee stings, right? And if they get stung or exposed to that peanut or bee sting, they can experience a system-wide very, very severe response that might make it hard for them to breathe and their heart rate slows down, et cetera, right? So when these molecules were first described, researchers realized that these signaling molecules play a role in that type of system-wide response, right? So that's why they were given the term anaphylatoxins. And so in order for other cells to respond to these anaphylatoxins, I think you shouldn't be surprised to realize, right, that these other cells need to have receptors on their surface that bind to these anaphylatoxins and use them as a message for that cell to now do something different, right? Like maybe get activated and contribute. So other phagocytic cells have these anaphylatoxin receptors. Endothelial cells, right? So the cells that actually make up the lining of the inside of your blood vessels have these receptors as well. That's why part of an anaphylatoxic shock, right, is that this smooth muscle that surrounds your blood vessels oftentimes will respond, right? They'll actually relax and, well, in some cases relax like around your blood vessels, right? So that can really suddenly cause a dangerous drop in your blood pressure. But other smooth muscle around things like your airways, they may do the opposite. They may actually restrict, right? Causing difficulty breathing. And mast cells respond to anaphylatoxins. We'll learn about mast cells later, but they're another example of innate effector cells. Right? And so, as we said, these anaphylatoxins induce these types of physiological changes down here. Okay. All right, last slide. So this is just to kind of summarize that, right? So the anaphylatoxins, let's pretend that you have a blood vessel here in cross section and it's surrounded by tissue. Maybe this is tissue that's your skin, right? Under your living skin that's underneath your outer epidermis. So if there's an infection in the skin, then the C3a and the C5a, they reach things like the endothelial cells that make up your blood vessels. Okay? And as a result, they cause, right, the blood vessel walls to relax and become more permeable. So as a result, the blood that was flowing through here contains lots of complement proteins and other immune cells. Right now, complement proteins can spill out into the tissue in greater amounts, right, to hopefully help out opsonizing the bacteria. Here's the bacteria hanging out here. Oh, this is terrible. This thing's drawn so badly to scale. It's not even funny, right? Because look, the bacteria are made to look like they're a third the size of a complement protein, right? When in reality, a complement protein is like 1/10,000th smaller than an individual bacteria, right? Okay, but anyhow, don't get me started on that. But at the same time, by weakening the walls of the blood vessel, entire cells like monocytes and neutrophils can also squeeze out, right, and begin to help out with the infection. Hopefully, this means that after another day or two of the induced, right, immune response involving these recruited factors, hopefully that'll be enough to take care of the infection. And 99.9% of the time, right, that combination of the immediate response for the first few hours, plus the additional recruited response takes care of our common everyday infections. So you don't even think about it anymore, right? And of course, if this doesn't work, then we're going to move on and talk about your adaptive response having to be woken up. All right. Okay, so that's it for the lecture today. So quick reminder, don't forget, finish your inquisitive homework assignments by tonight, right? Look for the quiz 1, which should automatically activate in Laulima in a few minutes at 1:30, right? That's due tomorrow night, right? So get into that routine of how these assignments work from week to week. And I'll look forward to seeing you on Friday. Right. Okay, I'll go to the chat now for any of you who want to stick around and see what kind of questions there are. All right, so Conrad asked a question. Is CD59 in a healthy cell embedded in the membrane or floating around? Right, so CD59 is a membrane anchored protein. So in other words, all the cells in your body, as part of the many thousands of genes that they're constantly expressing just to take care of normal housekeeping, being a cell kind of thing, right? It includes the CD59 protein. And that CD59 protein then is embedded in the membrane so that if you end up having accidentally a membrane attack complex forming, right, from some immune activity that's happening nearby, right, that the CD59, right, has the opportunity to bind and prevent the actual pore from taking shape. Okay, yeah, good question. All right, and that's the same for many of the other proteins that we looked at in the previous lecture that have to do with regulating things like improper C3b fixation on the surface of our normal healthy cells, right? We have proteins that also contribute with interfering with improper opsonization of one of our body's own cells by our own immune system. Okay. All right, good. Okay, so maybe that's it for questions. Let me scroll down. Okay, I think that's it. All right, good. All right, so I will see you all on Friday. Have a good rest of your week.